to Color Platform. Igniting the Curious Mind has on date 8 June 2026. Let's begin the chapter. Photosynthesis in higher plants. All animals, including human beings, depend on plants for their food. Have you ever wondered from where the plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, or other organisms depends on them for the needs. The green plant make and rather synthesize the food they need through photosynthesis and therefore called autotrophs. You have already learned that autotrophs kick nutrition is found only in plants, and all the organisms that depends on the green plants for food are heterotrophs. Green plant carry out photosynthesis, a physiochemical process by which uses light energy to derive synthesis of organic compound. Ultimately, all the living forms on Earth depends on the sunlight for the energy. The use of energy from sunlight by plants doing photosynthesis is the basics of life on the earth. Photosynthesis is important due to two reasons. It is primarily source of all the food on the earth. If it is also responsible for release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there was no oxygen to breathe? This chapter focuses on the structures of photosynthetic machineries, the various reactions that transform light energy into chemical energy. What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiment may have done in the early classes have shown the chlorophyll, a green pigment of leaves, lights and carbon dioxide are required for photosynthesis to occur. You have carried out an experiment to look for a starch formations in two leaves, variegated leaves, or a leaf that has partially been covered with a black paper. Exposed to light on testing these leaves for the presence of starch, it was clear that photosynthesis occur only in the green plants or on the leaves in the presence of lights. Another experiments have carried out where the part of a leaf is enclosed in a test tubes containing KOH, so cotton which absorbs carbon dioxide, while the other half exposed to air. The setup is then placed in a light for some times. On testing for the presence of starch, later in two parts of the leaf, you have found that the exposed part of the leaf tested positive for starch, while the portions that was in the tube and tested negative. This shows that carbon dioxide was required for photosynthesis. Can you explain for the reason and conclusion in the drawn? Earlier experiments. It is interesting to learn that the simple experiment that led to a gradual development in our understanding of photosynthesis. Joseph Priestley in 1733 to 1804 in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley may recall the discovery of oxygen in 1774. Priestley observed that the candle burning in a closed space, a bell jar, soon get extinguished. Similarly, a mouse would soon suffocate in a closed place. He concluded that the burning candle or an animal that breathed the air, both shows damages the air. But when they placed a mint leaves in the small bell jar, he found that the mouse stayed alive and the candle could continue to burn. Priestley hypothesized as follows. Plants restore to air that whatever breathe animals and burning candles remove. Can you imagine how Priestley could have conducted the experiments using a candle and a plant? Remember, we would need to rekind the candle to test where it burns in after few days. How many different ways can you think to light the candle without disturbing the setup? Using a similar setup as once used by Priestley, but by placing it once in dark and once in sunlight. John Egginghaus in 1730 and 1799 showed that sunlight is essential for plant process, that somehow purifies the air fouled by the burning candle or breathing animals. Egginghaus in England elegant experiment that aquatic plants shows that in bright sunlight, small bubbles were found around the green plants. while in the dark they did not. Later he identified the bubbles to be oxygen. Hence he showed that it's only the green plant that use could release oxygen. It is not until in 1854 that Julius Van Schans provided evidence of production of glucose when plants grow. Glucose is usually stored as starch. His latest study shows that the green substances in the plant's chlorophyll, as we know it now, is located in a special bodies which later is called chloroplast and within the plant cells. He found that the green plants in the parts in the plant as where glucose is made, and its glucose is usually stored in starch. Now consider the interesting experiment done by T.W. Eltman in 1843-1909 using a prism. It splits light into a spectral component and then illuminated a green algae, Cladophora, placed in a suspensions of aerobic bacteria. The bacteria were used to detect the sites of oxygen evolutions. He observed that the bacteria accumulated mainly in the regions of blue and red lights of the split spectrum. A first action spectrum of photosynthesis was described. It resembles roughly the absorption spectra of chlorophyll A and B. By the middle of 19th century, the key features of the plant photosynthesis were known. Namely, the plants could use light energy to make the carbohydrates from carbon dioxide and water. The empirical equations represented the total process of photosynthesis for oxygen evolving organisms has been understood as carbon dioxide, H2O light, H2O plus O2, where H2O represent a carbohydrate, glucose, and 6-carbon sugar. A milestone contributions to the understanding of the photosynthesis was made by the microbiologist Colin Van Neel in 1897 to 1985, who, based on his studies of the purple and the green bacteria, demonstrated that the photosynthesis essential are light-dependent reactions in which hydrogen forms a suitable oxidizable compound, reduces carbon dioxide to carbohydrates, which can be expressed by 2H2A plus O2 giving you 2A CH2O plus H2O. In the green plant, H2O is a hydrogen donor and is oxidized to O2. Some organisms do not release oxygen during photosynthesis. When H2O is instead of hydrogen donor for a purple and a green sulfur bacteria, the oxidation product is sulfur and sulfur depending on the organisms and not O2. However, hence he inferred that oxygen evolved by the green plants comes from H2O, not from carbon dioxide. This was later proved using the radioisotopic technique. The current equation that would represent the overall process of photosynthesis, therefore, 6CO to 12H2O, C6H12O6 plus 6 H2O plus 6O2, where C6H12O6 represents the glucose O2 released from the water, which was proved using radioisotope technique. Note that this is not a single reaction but a description of multiple step process called photosynthesis. Can you explain why 12 molecules of water as a substance are used and equations given above? Where does photosynthesis takes place? You would of course answer in the green plants or in the chloroplast based on what you really read in the chapter 8. You are definitely right. Photosynthesis takes place in green plants, green leaves of plants, but it does not so also in other green parts of the plants. Can you name some of the other parts where you can think of photosynthesis occurs? You have recollected the previous units that the mesophyll cells in the leaves have a large number of chloroplasts. Usually chloroplasts align themselves along the walls of mesophyll cells such that they get the optimum quantity of incident lights. When do you think the chloroplasts will be aligned with their flat surface parallel to the walls? When would they be perpendicular to the incident lights? You have studied the structure of chloroplasts in chapter 8 when the chloroplast, there is a membranous system consisting of grana, the stromal lamellae, the matrix stroma, where there is a clear division of labor within the chloroplast. The membrane system is responsible for trapping light energy and also for synthesis of ATP and NADPH. In stroma, the enzymatic reaction synthesis sugar, which in turn forms starch. From a set of reactions, since they are directly light driven by light reactions, the photochemical reaction, The latter is not directly light driven but are dependent on the products of the light reactions ATP and NADPH. Hence, to distinguish the latter, they are called by the convention of a dark reactions. A carbon reactions, however, this do not constituted to mean that they occur in darkness or they are not light dependent. Outer membrane, inner membrane, stroma lamellae, grana, stroma, ribosome, starch granules, and lipid droplets have many types of pigments are involved in photosynthesis. Looking at plants, have ever wondered why have there were so many shades of greens in their leaves even in the same plants? We can look at the answer to this question. We try to separate the leaf pigment of any green plants through a paper chromatography. Chromatographic separations of a leaf pigment shows that the color that we see in the leaves is not due to the single pigment but due to four pigment: chlorophyll A bright to blue green in chromatogram, a chlorophyll B yellow green, and xanthophyll yellow, carotenoid yellow to yellow orange. Let us now see what roles plays a various pigment plays in photosynthesis. Pigments are substances that are ability to absorb light at specific wavelength. Can you guess which is the most abundant plants pigment in the world? Let us study the graph shown ability of a chlorophyll A pigment to absorb a different wavelength. Of course, you are familiar with the wavelength of visible spectrums of light has such as a vigor. In the figure 11.a, you can determine the wavelength of a color of light at which a chlorophyll A shows a maximum absorptions. Does it show another absorption peak at any other wavelength too? If yes, which one? Look at the figure showing the wavelength at a maximum photosynthesis occurs in a plant. Can you see that the wavelength at which there is a maximum absorption by chlorophyll A in the blue and red region also shows a higher rate of photosynthesis? Hence, we can conclude that the chlorophyll A is a chief pigment associated with a photosynthesis. But by looking at the figure 11.c, can you say that there is a complete one-to-one overlapping between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? These graphs together shows that most of the photosynthesis takes place in blue and red region of the spectrum. Some photosynthesis does not this happen to a chlorophyll as a major pigment responsible for trapping light. Other thylakoid pigments like chlorophyll A, xanthophylls, carotenoids, which are called as accessory pigments, also absorbs lights and transfers the energy to chlorophyll A. Indeed, they are not only enable to a wider range of wavelength of incoming lights to utilize for photosynthesis, but also protect chlorophyll A from photooxidations. What is light reaction? A light reaction or a photochemical phases includes light absorptions, water splitting, oxygen release, and the formation of high energy chemical intermediates ATP and NADPH. Several protein complex are involved in the process. Pigments are organized into two discrete photochemical light harvesting complex within the photosystem 1 PS1 and photosystem PS2. These names in the sequence of their discovery, not in the sequence in which they are found during the light reactions. The LHs are made up of hundreds of pigment molecules bound to proteins. Each photosystem has as all the pigment except one molecules of chlorophyll A, forming a light harvesting systems also called antenna. These pigments helps in making the photosystem more efficient by absorbing different wavelength of lights. A single chlorophyll A molecules form a reaction center. The reaction center is different from both photosystem. In PS1, the reaction center chlorophyll A has an absorption peak of 700 nanometer, and there is a P700 which is P2, it is absorption of 680 nm, and that is nanometer, it's called as P680, the electron transport. In the photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electron to excite and jump into the orbit rather than atomic nucleus. These electrons are picked up by the An electron acceptor which passes through an electron transports. The system consists of a cytochrome. The movement of electron is downhill in terms of oxidation reduction or the redox potential scales. The electrons are not used by as they pass through the electron transport chain but are passed onto the pigments of photosystem 1 PS1. The photo the simultaneously electrons in the reaction center PS1s have also excited, but when they receive the red light of wavelength 700 nanometer are transferred to another acceptors molecule that have a greater redox potential. These electrons are then moved downhill again. This type of molecules of rich energy rich NADP plus and the additions of these electrons reduces NADP plus and NADPH and H plus. This whole scheme of transfer of electrons starting from the PS2 uphill to the electron acceptor down the electron transport chain of PS1, excitation of electron transfer of another acceptor finally downhill to NADP plus reducing it to NADPH and H plus is called as Z scheme. Due to this characteristic shape, the shape is formed when all the carriers are placed in a sequence of redox potential states, splitting of light. You have then asked, how does PS2 supply electron continuously? The electron that were moved from PS2 must be replaced. This is achieved by electron available due to splitting of water. The splitting of water is associated with PS2. The water is split into 2H plus O and electrons. This creates an oxygen, one of the net product of photosynthesis. The electron need to replace those and removed from the photosystem 1 are provided by photosystem 2. We need to emphasize here that the water splitting complex is associated with PS2, which itself is physically located on the either inner sides of the membrane of the thylakoids. When there are protons and O2 formed likely to be released in the lumen on the outer sides of the membrane, the cyclic and non-cyclic photophosphorylations. Living organisms have compatibility if of extracting energy from oxidable substance stored in the form of bond energy. Special substances like ATP carry this energy in the chemical bonds, the process through which ATP synthesized by cells in the mitochondria and the chloroplast is named photophosphorylation. The photophosphorylation is a synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in series, the photosystem 2 and the photosystem 1, a process is called non-cyclic photophosphorylations occur. The two photosystems are connected with an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH and H plus are synthesized by this kinds of electron flow. When only PS1 is functional, the electron is circulated within the photosystem and phosphorylation occurs due to cyclic flow of electrons. A possible location where this could happen is the stroma lamellae. When the membrane or the lamellae of the grana have both PS1, PS2, stroma lamellae member lacks PS2 as well as NADPH reductase enzymes. The excited electron does not pass on to NAD plus but is cycled back to PS1 complex through electron transport chain. The cyclic flow hence results in the synthesis of ATP and not NADPH and H plus. The cyclic photophosphorylation is also occurs when only light of wavelength beyond 680 nanometers are available for excitation. Chemoosmotic hypothesis. Let us now try to understand how actually ATP synthesized in the chloroplast. The chemiosmosis hypothesis was being put forward to explain the mechanisms. Like in respiration, photosynthesis too, ATP synthesis is linked to development of proton gradient across a membrane. This time these are membrane thylakoids. There is no difference through they are proton accumulation towards the insides of the membrane in the lumen. In respiration, the protons are accumulated in the intermembranal space of the mitochondria where the electrons move from ETS. Let us understand the cause of the proton gradient across the membrane and we need to consider again the process that takes place during the activation of electron and their transports to determine the steps that causes a proton gradient to develop. Since the splitting of water molecules takes place on the inner sides of the membrane, the proton or the hydrogen ions that are produced by the splitting of water accumulates within the lumen of the thylakoids. As the electron moves to the photosystem, the photons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers the electron not to the electron carrier but to the H carrier. Hence, the molecules moves a proton from the stroma while they're transporting an electrons. When the molecule passes on its electron to electron carrier on the inner sides of the membrane, the photon is released into the inner sides of the lumen of the membrane. The NADPH reductase enzyme is located on the stroma sides of the membrane. Along the electron that comes from the acceptor to the electron PS1, the photons are necessary for the reduction of NADPH to NADPH and H+. These photons are also removed from the stroma. Within the chloroplast, photons in the stroma decreases in number, while in the lumen there is accumulation of proton. This creates a proton gradient across the thylakoid membrane, as well as a measurable decreases in pH in the lumen. Why we are so interested in the proton gradient? This gradient is important because it's a breakdown of a gradient that leads to the synthesis of ATP. The gradient is broken down due to movement of proton across the membrane to stroma through a transmembrane channel, that is CF0 of ATP synthesis. The ATP synthesis enzyme consists of two parts. One called CF0 is embedded in the thylakoid membrane and forms a transmembranal channel that carries off oscillation diffusion of proton across the membrane. The other proton is called CF1 and the protrude of the other surface of the thylakoid membrane on the sides of the faces the stroma. The breakdown of the gradient provides enough energy to cause the conformational changes in CF1 particles of ATP synthesis, which can synthesize enzymes, several molecules of energy-packed ATP. The chemiosmosis requires a membrane, a proton pump, a proton gradient, ATP synthesis. Energy is used to pump proton across a membrane to create a gradient or a high concentration of protons within thylakoid lumen. ATP synthesis has a channel that allows the diffusion of protons back across the membrane. This releases enough energy to activate ATP synthesis energy that catalyze the formation of ATP. Along with the NADPH productions, with the movement of electrons, the ATP will be used immediately by the biosynthesis synthetic reactions taking place in the stroma, responsible for fixing CO2 and synthesis of sugar. Where are ATP and NADPH used? We have learned in the products that light reactions are ATP, NADPH, O2. Of these, oxygen diffuses out of chloroplast, while ATP and NADPH are used to drive the process leading to synthesis of food, more accumulate sugar, and this is a biosynthetic process of photosynthesis. This process does not directly depend on the presence of light but is dependent on the product of the light reactions, that is ATP and NADPH, besides carbon dioxide and H2O. You have wondered how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic process continue for some time and then stop if then the light is made available. The synthesis starts again, and we can hence say that the calling the biosynthetic phases as a dark reaction is a misnomer. Discuss it amongst yourself. Let us see how the ATP and NADPH are used in biosynthetic phases. We saw earlier that carbon dioxide is combined with the H2O to produce CH2O2. CH2O in the bracket here of a sugar. It was interesting to the scientists to find out how this reactions proceed, or rather what's the first product formed when a carbon dioxide is taken into the reactions of fixed. Just after World War II, among the several efforts have to put for radioisotopes beneficial uses, the work of Melvin Kelvin is a exemplary. The use of radioactive carbon-14 by him in the algal photosynthesis study led to a discovery of carbon dioxide fixation product, which was 3 carbon organic acids. It was contributed to work out complete biosynthetic pathway. Hence, it's called as Calvin cycle for him. The first product identified has three phosphoglyceric acids, and in short, PGA. How many carbon atoms does it have? Scientists have also tried to know where or whether all the plants have PGA as the first product of carbon dioxide fixation, or whether any other products have been formed in other plants. Experiments conducted over a wide range of plants led to a discovery of another group of plants where the first stable product is carbon dioxide fixation was again an organic acid, but one had four carbon atoms in it. This acid was identified as oxaloacetic acid or OAA since oxygen carbon dioxide assimilation during photosynthesis was also said not to be a main types. These plants in which was first a product of carbon dioxide fixation of C3 acids PGA, that is C3 pathway. This was where the first product was C4, that is C4 acid OAA, and in the C4 pathway. These first two group of plants show a both association of characteristics that we have discussed later. The primary acceptor of carbon dioxide. Let us now ask ourselves a question: what was asked by a scientist who was struggling to understand the dark reactions? How many carbon atoms would a molecules have after accepting carbon dioxide? Would have three carbons. The studies were unexpectedly shown that the acceptor molecules of 5 carbon keto sugar ribulose bisphosphate did any of you think of this possibility? Do you worry? The scientists also took a long time. Don't worry, scientists also took a long time to conduct many experiments to reach these conclusions. It was believed that since the first product was C3 acids, a primary acceptor which is a 2 carbon compound, they spent many years trying to identify the 2 carbon compound before the discovery of 5 carbon RUBP. The Calvin cycle. The Calvin and his co-worker worked out the whole pathway, showed that the pathway operated in a cyclic manner. RUBP was regenerated. Let us now see the Calvin pathway operates, where the sugar is synthesized. Let us now outset the understanding very clearly. The Calvin pathway occurs in all photosynthetic plants. Does it not matter whether they are C3 or C4 pathway? Easy of understanding, Calvin cycle can be described under 3 stages: carboxylations, reductions, and regenerations. Carboxylation is a fixation of carbon dioxide into a stable organic intermediate. The carboxylations is the most crucial steps in Calvin cycles, where carbon dioxide is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzymes RUBP carboxylase, which results in the formation of 2 molecules of 3-PGA. Hence enzymes has an oxidation activity. It occurs in more correct way to call it as a rubisco bisphosphate carboxylase oxygenase or rubisco. Rubulose 1,5 bisphosphate atmosphere carbon dioxide H2O carboxylation 3 phosphoglycerate reduction ATP plus NAD giving you ADP plus PI plus NADP plus, and triphosphate sucrose to starch, and then which moves to regeneration giving out AD from ATP to ADP and then back into ribulose 1,5 bisphosphate. The reductions. These are a series of reactions that lead to formation of a glucose. The first step involves utilization of 2 carbons of ATP for the phosphorylation and 2 NADPH for the reduction of CO2 molecule fixed. The fixation of 6 molecules of carbon dioxide and 6 molecules turns of cycles are required for the formation of 1 molecules of glucose from the pathway. The regeneration of the carbon dioxide acceptor molecules RUBP is crucial in the cycle is to continue uninterrupted. The regeneration steps requires ATP for the phosphorylations to form RUBP. Hence, for every CO2 molecules entering a Calvin cycle, 3 molecules of ATP and 2 NADPH are required. It is probably to meet these in the number of ATP and NADPH used in the dark reactions that a cyclic phosphorylation takes place. To take a 1 molecules of glucose 6 turns in a cycles are required. Work out how many ATPs and NADPH molecules will be required to make 1 molecules of glucose to the Calvin pathway. It might help to understand all, but if we look at what goes in and what comes out of Kelvin cycle, in and out 6 CO2 and 1 glucose, 18 ATP and 12 NADPH, giving out 18 ADP and 12 NADP. C4 pathway. Plants are adapted to dry tropical regions have C4 pathway mentioned earlier. So these plants have C4 oxalic acids has a first carbon dioxide fixation product. They uses carbon C3 pathway or the Calvin cycle has a main biosynthetic pathway in which they may differ from C3 plants. This is a question that you may be reasonable to ask. C4 plants are special. They have a special type of leaf anatomy. They tolerate higher temperature. They show a responsible to high light intensity, and they lack a process called as photorespirations, have a great productivity of biomass. Let us understand these one by one. Study vertical sections of a leaves. One of a C3 plants, that of a C4 plant. Do you notice any differences? Do both have a same types of mesophils? Do they have a similar cells around the vascular bundles? A particularly large cells around the vascular bundles of C4 plants are called bundle sheath cells. The leaves which have such anatomy are called as crans anatomy. The crans means width and they are reflections of arrangement of cells. A bundle shell sheath cells has several layers around the vascular bundles, and they are characterized by having a large number of chloroplasts. Thick wall imperversions of a gaseous exchange are known into cellular spaces. You may like to cut a section of leaves of C4 plants, a mason sorghum, to observe the cross anatomy and distributions of a mesophil cells. It'd be interesting for you to collect leaves of a diverse species of plants around you and cut the vertical section of the leaves. Observe under the microscope. Look at the bundle sheaths around the vascular bundles. The presence of bundle sheath would help you to identify the C4 plants. Now, study the pathway shown in the figure 11.9. The pathway that has been named as a Hatch and Slugs pathway. It's again a cyclic process. Let us study the pathway by step. Listing the step. Primary CO2 acceptor is 3 carbon molecule phosphoenol pyruvate PEP, and this is present in the mesophil cell, and the enzyme is responsible for fixations of PEP carboxylase or PEP cases. It's important to register that the mesophil cells lacks rubisco enzymes. The C4 acids, oxaloacetic acid, is formed in the mesophil cells. It is then from other 4 carbon compound like malic acid, aspartic acid in the mesophil cells itself, which is transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release CO2 and 3 carbon molecules. The carbon molecule is transported back to the mesophil, where it has been converted in P to PEP again, and thus completing the cycle. CO2 is released in the bundle sheath cells, entered C3 and the Calvin pathway, the pathway common to all plants. The bundle sheath cells are rich in enzyme rubisco bisphosphate oxygenase carboxylase oxygenase. Rubisco put lacks of PEP cases, but the basic pathway results in the formation of sugar. Calvin pathway is common in C3 and C4. Did you note that the Calvin pathway occurs in all the mesophil cells of C3 plants? In the C4 plant, it does not take place in the mesophil cell, but does so in the bundle sheaths. The photorespiration. Let us try to understand one more process that creates an important differences between C3 and C4 plant, that is photorespirations. To understand the photorespiration, you have to know a little bit more first about the first steps of Calvin pathway. The first, carbon dioxide fixation steps. It's a reaction of RUBP rubisco RUBP combines with carbon dioxide to form 2 molecules of 3-BGA, that is catalyzed by rubisco. RUBP plus CO2 plus Rubisco to 3-PGA and Rubisco, that is four most abundant enzymes in the world. It is characterized by the fact that there is an active site that bind to carbon dioxide and oxygen, hence the name. Can you think of how this could be possible? The Rubisco has a much greater affinity for carbon dioxide and when the carbon dioxide is to O2 is really equal. Imagine what would happen if they were not so. This binding is competitive. It is relatively concentration of carbon dioxide and the carbon dioxide that determines which of the two binds to the enzymes. The C3 plant shows that some oxygen binds to Rubisco and hence carbon dioxide fixation is decreased. Hence, the Rubisco instead of being converted to two molecules of PGA bind to O2 to form one molecules of a phosphoglycerate and phosphoglycolate. Phosphoglycolate to carbon in a pathway called as photorespiration. In the photorespiration pathway, there is neither a synthesis of sugar nor ATP. Rather, it's a result of a release of carbon dioxide with a utilization of ATP. In the photorespiration pathway, there is no synthesis of ATP and NADPH, and a biological function as photorespiration is not known yet. In the C4 plants, the photorespiration does not occur, and this is because the mechanisms that are necessary for concentration of carbon dioxide at the enzyme sites takes place in C4 acids from the mesophyll is broken down in a bundle sheet cells release carbon dioxide. This results in increasing of intracellular concentration of carbon dioxide. In turn, this ensures that the Rubisco functions as in carboxylase, minimizing the oxygenase activity. Now, what you know that the carbon dioxide C4 plants lacks photorespirations, you probably can understand why the productivity yield are better in these plants. In addition, the plants show a tolerance to high temperature. Based on the above discussions, you can compare the plants shows the C3 and C4 pathway. Using the table given, fill the information below. The factor affecting photosynthesis. An understanding of the factor that the effect of photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of the plant, including crop plants. Photosynthesis is under the influence of several factors, both internal plant and external. The plant factor includes the number, size, age, orientation of leaves, mesophyll cells, chloroplast, internal CO2 concentration, and amount of chlorophyll. The plants or the internal factor are dependent on the genetic predisposition and the plant's growth of the plant. External factor would include the availability of sunlight, temperature, CO2 concentrations, and water. As a plant photosynthesis, all these factors will simultaneously affect its rate. Hence, the several factors interact simultaneously affects photosynthesis carbon fixations. Usually, one factor is a major cause or is one that limits a rate. Hence, at any the rate will determine the factor available at the suboptimal level. When several factor, any biochemical process, Blackman's law of limiting factor comes into effect. This states that the following: if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is a factor which directly affect the process by its quality its change. For example, despite the presence of green leaf at optimal lights and CO2 conditions, the plant may not photosynthesize if the temperature is very low. This leaf, if we given the optimal temperature, will start the photosynthesis. Light, we need to distinguish between the light quality and light intensity, duration of exposures of light. While discussing light has a factor that affect the photosynthesis, there is a linear relationship between the incident lights and the carbon dioxide and fixation rate at the low intensities. The light intensities gradually rate does not show further increases as other factor affecting limits. What is interesting is to note that the light saturation concentrations occurs in 10% of sunlight. Hence, except for plants and shades or in a dense forest, the light is really a limiting factor in nature. Increase in the incident light beyond a point causes a breakdown of chlorophyll and a decrease in photosynthesis, carbon dioxide concentration. It's a major factor limiting photosynthesis. Concentration of CO2 is very low in atmosphere below 0.03 to 0.4%. Increase in concentration of 0.5% can cause an increase in carbon dioxide fixation states. Beyond this level can be damaging for over a longer period. C3 C4 plants respond differently in carbon dioxide concentrations at a low in light conditions. Either low group responds to a high CO2 condition. At the high intensity, both C3 C4 plants show an increase in rates of photosynthesis. What is important to note is that the C4 plant show a saturations at C360 mul, while that C3 plant shows increase in carbon dioxide concentration saturation is below 420 mul. Thus, the current availability for CO2 level is limiting to the C3 plant. The fact that C3 plants responds to our CO2 concentrations by showing an increased rate of photosynthesis leading to a higher productivity has been used for some greenhouse crops. House crops such as tomato, bell peppers, and also heavier grown in a carbon dioxide enriching atmosphere that leads to a higher yields. Temperature, the dark reactions, enzymes, enzymatic temperature control through a light reaction, also temperature sensitive that affect to a lesser extent. The C4 plants respond to a higher temperature and show a high rate of photosynthesis, while 3 plants show a much lower temperature optimum. The temperature optimum for photosynthesis at different plants also depends on the habitat as they are adapted to. Tropical plants have a higher temperature optimum than that of a plant adapted to temperate climates. Even though water is one of the most reactant in the light reactions, the effects of water as a factor is more, though its effects on the plants rather than directly on the photosynthesis. Water stress causes tomato to close, hence reducing the CO2 availability. Besides, the water stress also takes wilts and thus reducing the surface area of the leaves and the metabolic activity as well. Summary: the green plants make their own food of photosynthesis. During the process, carbon dioxide forms atmosphere is taken in the leaves through the stomata and used for the making carbohydrates, principally glucose, starch. Photosynthesis takes place only in the green plant parts and mainly in the leaves. Within the leaves, the mesophyll cells have a large number of chloroplasts and responsible for carbon dioxide fixation, while the chloroplasts are membrane sites of light reactions, while the chemosynthetic pathway occur in the stomata. The photosynthetic has two stages: light reaction, carbon fixing reaction. In the light reactions, the light dependent is absorbed by the pigment present in the antenna. The funneled to a special chlorophyll A molecules reaction center. There are two photosynthetic systems, PS1, PS2. PS1 is 700 nanometer absorbing chlorophyll of PA 700 molecules in the reaction center. PS2 has a 680 reaction center that absorbs right reaction at 680 nanometer. After absorbing light, the electrons are excited and transferred to PS2 and PS1. Finally, NAD forming a NADH. During the process, a proton gradient is created across the membrane of the thylakoid. The breakdown of proton gradient due to a movement through F0 parts of ATP enzymes releases enough energy for synthesis of ATP. Splitting of water molecules is associated with the PS2, resulting in release of O2 proton and transfer electron of to PS2. In carbon fixation, CO2 is added to the enzymes rubiscoase to the 5-carbon compound RUBP to convert 2 molecules of 3-carbon PG, and this is converted to a sugar by the Calvin cycle. The RUBP is regenerated. During the process of ATP and ADP synthesis in the light reactions are utilized. RUBisco are catalyzed by the wasteful oxygenation reaction by C3 plants of photorespiration. Some tropical plants such as a special type of photosynthesis called C4, and in these plants the first product of carbon fixations takes place in a mesophyll in a carbon 4-carbon compound. In a bubble sheet cell called as Calvin cycle is carried out of synthesis for the carbohydrates. Thank you for watching. You're tuned into Column Platform Igniting the Curious Mind, where you and I grow together. Thank you.